Stanford University.
Wow, it's great to see everyone here, and what a great turnout for Hannah. Hannah's a woman who needs no introduction, but I'm going to introduce her anyway. And so we'll skip the early years. She did a lot of her early training in England, and then came over to the United States in 1985, I think, as a young, energetic cardiology fellow, and has risen up through the ranks very quickly here, through the academic ranks very quickly here at Stanford. Hannah was the first one to do a randomized controlled trial investigating a genomic test to diagnose human heart transplantation rejection, and that test today is used all throughout this country and indeed all around the world. And Hannah has gone on from that first experience to develop new technologies herself, and made a great collaboration with Steve Quake here at Stanford, and began to investigate the use of sequencing of circulating DNA to diagnose cardiac transplantation rejection. Hannah has another life, in addition, and in 2004 she became the Senior Associate Dean for Diversity and Leadership here at Stanford. And so Hannah has continued to be a very solid member of the transplantation team until 2014, when we loaned her to the NHLBI. And she went there to assume a couple of titles. One was the title as the first NIH Chief Officer for Scientific Workforce Diversity, as well as being a senior investigator in the internal program at the NHLBI. So we're extremely happy to have her back visiting from the NHLBI, and it's been a pleasure to get to meet with her yesterday and today. So please welcome Hannah.
Tom, thank you very much for that lovely introduction. What Tom didn't tell you was that, in fact, he was the one that developed the tools for the gene expression profiling so that it could actually be ultimately tested in heart transplant. So for that, we really recognize your support. And I think it speaks to the importance of this interdisciplinary work that is needed to be done in order to rapidly move new ideas into the clinic. And so what I'm going to talk to you about is how I'm viewing this new approach, these new genomic tools, to bring it to the clinic, not only for heart and lung transplantation, but for all solid transplantation. And wouldn't it be wonderful if ultimately we could develop here at Stanford a center for transplant genomics that could drive this work that would be spanned from basic right through to delivery. So my talk is going to first present a case. Really, I do that for you to get a vivid picture of what the phenotypes that we are talking about, that we want to better detect and predict early, and so that you can see how those phenotypes ultimately affect the lives of our patients. I'll then talk about the cell-free DNA technology itself and how it came about to be applied to transplantation in the first place, and then tell you where we are with it in the work that I've done since I left Stanford in trying to not only use it for the diagnosis of acute rejection, but develop technologies even further. So here we are. This patient is known to me and a few of my colleagues from the clinic that I see here, a delightful woman whom we met when she was age 29. In fact, she had undergone a transplant as a child. She had familial cardiomyopathy. Unfortunately, her brother died of the disease, and she and her twin sister were seen at the Texas Heart Institute, where they underwent heart transplantation, did extremely well. And we first met this patient when she relocated to the Bay Area with her husband, 3 months pregnant. And she came to our clinic to be looked after by myself and Sharon Hunt and others, and we were very excited with the opportunity of having to look after this pregnant woman who'd had a transplant. May I say that wasn't the first case. We'd had some experience in doing this, and we were all very happy that she did very well through the pregnancy, delivered pretty close to term, and appeared to be doing well until a few months after the delivery. She turned up in clinic one day and was clearly in heart failure. And to cut a long story short, what we ultimately discovered that she had mounted antibodies to the graft through antigens that were shared with the father. And so that way she had auto-sensitized herself, developed antibody-mediated rejection that led to progressive transplant coronary disease and ultimately the need for transplantation. So let me just show you some of the images that we collected. Here she was. This is the echocardiogram, which shows that she had a fairly normal LV function. Unfortunately, it's not projecting very well, but on the computer you can see very nicely the endocardium coming in nicely. And then what I'm trying to show you here is that very time when later on she develops this really quite poor ventricular function with an ejection fraction of 25%. Much to our dismay, we're very anxious about it. We do biopsies, find only mild cellular rejection there, and then do some certain antibody tests, donor-derived antibody, and discover that she has mounted a B-cell immune response to the graft. And here is her coronary angiogram. And again, unfortunately, the lighting is not tremendous here, but what is showing here is this typical vascular disease that we see in the transplant patients. There we go, thank you. With this peripheral pruning, you see there's normal vasculature here, and it just gets out, mouth-eaten look out there. And this was the reason why she was in congestive heart failure. And thankfully, we looked after her well, put her on the retransplant list, and within a few weeks, she got a donor and was retransplanted. I'm showing you this just to show you again that this disease, when we talk about the phenotype of the allograft vasculopathy, we're not only going to be thinking about what is happening on the coronary angiography, but what happens a lot earlier in terms of the intimal proliferation that occurs. And thankfully, we're now able to see that way ahead of time with this intimal thickening. So that's what we aim to diagnose early, to prevent, and to treat. So back to our patient, she had her transplant, and there she is. There she is with her baby now a year after the retransplant. And of course, there she is just last year as a rambunctious 8-year-old at the transplant reunion party. And you can see, so this is what drives physicians like myself, physician scientists. There's experiences of what we see in our patients to actually try to take those observations back to the laboratory and figure out how we can come up with testing and therapy to make the outcomes even better. Talking about outcomes, here is a slide that just shows you what the expectation currently is for a heart transplant patient. So what you're showing here is that on average, the median survival is 11 years, and half of the patients will be alive at 10 years, doing extremely well, but we are plagued with these complications, the allograft vasculopathy, acute rejection, and other complications that we seek to understand better. One of the major ones is acute rejection, and throughout the years, when I got here in 1985, the use of cyclosporin as the major immunosuppression was already in place, And what that meant was the acute rejections that we saw in the patients were less dramatic. And so we had to find another way of diagnosing it more rapidly to actually be able to treat it. And so this procedure called the endomyocardial biopsy came into play, which is, for those of you who have seen this, you put a catheter into the internal jugular vein, and if you're lucky, you get a pinhead-sized piece of endomyocardial tissue from the right ventricle and send it off to the pathology, and it's read and graded. The problem is that there are complications to it, and one of our surgeons once did this, and when the biopsy came back, not only was there endomyocardial tissue, but there was also stomach. So you can imagine where that catheter had gone. So it's true to say that there are complications, but they're not that common in good hands. But I think these are the difficult problems. You know, inter-observer variability for reading a biopsy is huge. It's almost like a toss of a coin. So that is problematic, and more on that, it is extremely expensive. So as Tom alluded to, we have been in the quest for non-invasive tests. Firstly, the work that I did was around echocardiography. That's what I did my thesis here when I came here in '85 with Sharon Hunt, John Schroeder, and many others, and showed that diastolic function was a sensitive marker, but it wasn't quite specific enough to lead to the ousting of the biopsy. And so fast forward, about 5 years ago, while I was still in the dean's office, I began to follow this field of cell-free DNA. And just a few facts for those of you who are not familiar with it. The fragments of DNA, and this is nuclear DNA, and I emphasize that because I'm going to be talking about DNA from another source in a moment. These may arise from apoptotic or necrotic cells from anywhere, and it could be endothelial cells, and that is another area of great research and interest that I think we can learn from as those technology comes in. That is to say, what is the cell type from which this is occurring? But what we have is that you can actually find fragments of cell-free DNA in all kinds of tissues, including plasma. They are short, only about 160 base pairs, and that has implications for the kinds of technology needed to measure them, and we'll talk about that in a moment. They could be double-stranded or single-stranded. Another technology that is developed focusing on the single-stranded type of assays that give us great yield. The nice thing about it, it has a half-life which is short, and so it really is indicative of injury and what's going on. So, as in most things, you think you have come up with a brand new idea yourself, and it hasn't been tested before. But as we looked into the literature, of course, we found that cell-free DNA had been used a fair bit in prenatal diagnosis, cancer biomarkers, autoimmune diseases, and various types of tissue injury. But what the transplantation model allows us to do is tip into the low-hanging fruit. That is to say, we know that two different genomes are present, and we have an opportunity for doing our work that will allow us to pick up what is from the donor and what is from the recipient, certainly as a percentage. So the first story around this begins, as I said, about 5-6 years ago. There I was sitting in the dean's office just up here, and I think I had a little moment to spend, and I was actually reading medical journals about science rather than diversity. And I saw this article that Steve Quake had published, which showed the non-invasive diagnosis of using cell-free DNA as for prenatal diagnosis to pick up trisomies and other chromosomal abnormalities, which, by the way, this work has really forestalled and taken off in the field, such that amniocentesis, which many people like myself needed to have because of having one's children late and literally sneaking under the biological time clock, people don't need to do that anymore. So I went to Steve, and we met at Clark's at the Clark Center at Pete's there, and I said, this sounds like a great idea. And Steve's reply to me was that, well, the organ transplant is essentially a genome transplant. And I was expecting and hoping to meet people like you here. He just arrived. And so the rest was history. We dug into some specimens, biorepository of specimens that I'd collected as part of a program project grant several years ago, reasoning that if we were looking for DNA, they should be okay, and did the proof of concept experiments, and it turns out it was quite correct that we were able to pick up donor-derived DNA. This work actually went on to be validated in a greater number of patients, 66, I believe, and in 500 samples. And this is the working with Kiran Kush here and others to bring that to publication. And along the way, we realized that the cell-free DNA was really not only just of human origin, that we were able to pick up microbial DNA as well in the process. And following the leads of that led us to publish this paper in Cell, where we were able to demonstrate very nicely that there's a change in the level of the virome. For example, rhinellovirus, as the patients become more immunosuppressed, you see more of it. And not only that, you could actually pick up certain unknown, previously identified viruses in the circulation. So that was a very exciting part of it. And then we took this further into lung transplantation, where the challenges, I would argue, are even greater than in heart transplantation, to demonstrate how you can simultaneously, on one sample, measure the cell-free DNA that is coming from the donor, that is microbial in origin, and then figure out what is the major player here, whether it's rejection or infection. So here is the technology in a nutshell. I think many of you who have followed this have seen this. It depends on SNP positions for which donor and recipients are homozygous within a single base pair, and that are present in both alleles. You remember the nuclear genome, two alleles. That allows for discrimination of donor and recipient. So imagine here a person, the red being shown as the recipient DNA, lots of it circulating. There are about 100 million fragments of cell-free DNA per mil of plasma. And imagine then you put in a transplant, denoted in green here, and as there is injury to the graft, you see fragments of DNA come out into the circulation that can be picked up by the technology that I've just described. And so what we have always been thinking, and I'll show you some of that a moment, is that the graft gets injured, perhaps during ischemia reperfusion, perhaps even before explantation, and that releases the DNA. And I'll show you data to show that, yes, in the first few hours, percent cell-free DNA is really high, and then it decays. But the other part of this that has really tweaked my attention recently, it has to do with the pro-inflammatory effects of DNA, naked DNA that is, and I'll show you that. And so I'm beginning to think, what is cause and effect? Is it that we are having high levels of donor-derived DNA circulating, and that is eliciting an innate immune response, which then sets the stage for the downstream effects later on? And that is going to be pivotal, a central hypothesis that I've been working on at the NIH. So here is the technology. that Steve's lab taught us how to do. You take blood, you genotype donor and recipient, collect plasma afterwards, do the shotgun sequencing, and you identify the reads for donor and recipient SNPs and calculate the % donor-derived DNA. And this work, as I mentioned, has been published, and here's just one example to show you that not only can you actually see levels of the DNA rise, or the percentage of the donor DNA. Remember, we are not measuring concentration. We might be able to get there at some point to measure concentration. This is the percentage of the donor-derived DNA that moves along and increases at times of, here is cellular rejection, patient gets treated, goes down, antibody-mediated rejection goes up again, and so forth. And in patients who have come to retransplantations, we see a plummeting down of the cell-free DNA. So we are actually confident that we are measuring something that is extremely real, but one swallow doesn't make a summer, as is said. Here's the sort of statistical analysis behind that, and I think you will agree that the moderate rejections have a relatively higher % donor-derived DNA compared to the patients who are not rejecting. And as is classically done, we want to know what the receiver operating characteristics are. That is to say, plotting the true positive rate, but the true positive rate versus the false positive rates. And if that was absolutely perfect, of course, as you all know, you'll get a straight line, which is 1. So, but an AUC of 0.8 is pretty darn good. And what we've shown is that it's the variability of the reading of the biopsy is what contributes to this lack of diminished in the sensitivity. So when we saw all that, we reasoned quite rightly that, of course, this could possibly be related to any organ transplant. And we were fortunate enough to get one of the ARA grants to actually study this in heart and lung transplant recipients. And Steve, as he's extremely astute. If those of you who don't know him should know, I was always pushing for this being a test that would be particularly helpful for heart transplant and probably not that helpful for lungs, because we always thought, oh, the lungs, they're very complicated anyway. There's infection, there's rejection. But actually, I think Steve was right in basically what we've seen. It starts off by knowing what the outcome for lung transplantation is, as shown in the slide. Worse, not nearly as good as the outcome after heart transplantation. So we have an even greater unmet medical need within the lung transplant to figure out why it is we're losing these grafts. And most importantly, to be able to pick it up early and do something about it. So we lose grafts, lung grafts, from bronchiolitis obliterans syndrome, which is this fibrotic and obstructive in the airways, in the small airways, that actually leads to loss of graft function. And in addition to airways damage, guess what? There is also vascular damage. I used to have a nice histology slide that shows that, so where you can see the intimal thickening occurring within the vasculature of the lung. So we are thinking, of course, that this combination of injury, infection, alloimmunity results in these poor outcomes. But of course, we wanted to ask the simple question, as in the hearts, whether or not this was a reasonable tool for recognizing rejection. And there it is. You can see there's a separation between those biopsies that are deemed as severe rejection with an AUC that is even stronger than that of the heart. So that's the first part of the story that takes us on to say that, yes, we have reasonable evidence that this test might be helpful. And of course, all of you who follow biomarker work, that is just the beginning. You need to then externally validate across platforms and across different patient samples, and you need to then be able to do this, to demonstrate the clinical utility, which is your new test really going to add more to what is already there? So let's switch tracks a little bit and talk about the infection story very briefly. I mentioned to you that along the way, we of course realized that we were looking at not only human DNA, but microbial-derived DNA, and you can see it here, and this was part of the report in Cell. And when we looked down, we could actually identify what this DNA, this microbial in origin was, relative to viral, bacterial, and so forth. And mostly, there was this, much of it was single-stranded DNA, which has relevance for technology, which I'll describe in a moment. I mentioned that the anellovirus was the one that was most abundant, appeared to move along with the level of the immunosuppression, and at that point we began to think, wouldn't it be interesting if in fact this could be an indirect marker of the relative immunosuppression of any given patient, given the fact that what we have right now, the blood levels of the drugs, is an extremely, extremely blunt instrument to measure the level of immunosuppression. So in that paper, we described that we could detect infection after lung transplantation, particularly CMV. CMV disease is a major, pathologic infection that was going on, and you can see it here that it was detected quite easily and with a really good AUC there. And in addition, as I mentioned, we were able to diagnose, to recognize previously undiagnosed infection. So this part of the story of infection, I think, becomes highly relevant for lung transplantation, but also for other transplants. Because after all, we recognize that in many instances, we have subclinical viremia going on that goes undetected. And there's a lot of the literature out there, to which I contributed, that indicates that subclinical viremia in itself is hugely pro-inflammatory, and that contributes to the development of the chronic outcomes. More about that later. So by this time, it's 2014, early spring, exactly about 3 years ago, and I get recruited to the NIH to lead the diversity efforts. But in discussing my recruitment, it was very clear that the expectation was that I would continue to do my research and do science. And actually, the advisory committee to the director, to the NIH director, was that this person who was to lead diversity needed to be a practicing scientist. And this is quite strategic because when you do that, you're living and breathing in the lives of the people whose hearts and minds you wish to change through the diversity work. And so that's how I did that. So I'm an investigator in NHLBI, but of course, when I got there, actually I knew this before I got there, but it's a better story to say that when I got there, I discovered that there's actually no organ transplantation at NIH Clinical Center. I don't know if any of you have visited the clinical center, beautiful, but that was part of the allure. Actually, you walk through this in this whole hospital that is just there for clinical research. It's just amazing. But no organ transplantation. So what to do? So I went cap in hand to Hopkins and talked to their people there and said, would you like to join this consortium, which we called the Genomic Research Alliance for Transplantation. I thought with the cardiologist's love for acronyms, I thought it was appropriate. We call ourselves GRAFT. And so we have the University of Hopkins, Maryland, Inova Fairfax, which is a private institution, Virginia Commonwealth, and MedStar Washington. So what happens is that they recruit patients, they run them through the protocols that we provide, collect specimens, and most importantly, collect the clinical data, which then gets curated and comes back to a database in my lab. And the first thing to note about this patient population is they are diverse. It's a diverse population. And we have been able to, I'll show you in a moment, look at so far who we've recruited into this essentially a natural history study. And you can see that there's a large number of women, proportion of women. And you also see that there is this population that is enriched for African American. And why do I say that? The reason I tell you that is because regardless of which organ transplant you look at, be it kidney, be it heart, be it lung, the outcomes following organ transplantation are poorer for non-Caucasian populations. And for African Americans per se, it used to be thought that it was due to medication compliance. Well, Karen Cush and myself did some work on a large image study, and I'll show you, that really, I think, fairly debunks that theory. And now we're thinking about what is the genetic basis of it. So in this GRAFT consortium, which began in 2015, we transferred the technology of the work here through the core laboratory there and then to my team in my lab. We have this ongoing enrollment from 5 different centers, and I've already mentioned the diversity of the population. We now have a biorepository of over 25,000 specimens and collecting all the other relatively important specimens that we can able to study immunologic pathways ultimately. So, so the research agenda for my lab is summarized here, and I'd like to take you through some of the examples of what we are doing to get you a sense of where this work is going in the intramural program at NIH. The first order of the day was obviously to transfer the technology from here to there to the laboratory. And then to look at what are the phenotypes that this marker can detect. Can it predict long-term outcomes? What are the genetic basis of the differences in solid organ transplantations? And that's where we get into the relevance of mitochondrial DNA, which quite frankly is extremely exciting to me, and then the infection part of this. So how did we do with the reproducibility studies? Well, when I got to NIH, I'm sure a lot of you might have been following that. There was a great big hoo-ha out there about the fact that so few studies are reproducible, particularly genomic studies. And there was a conference convened to set standards of how one should go about confirming reproducibility. And so the conference identified 3 steps, which we followed quite closely. One was to establish the local computational workflow, validate that workflow, and then use published files to reproduce the results. The second part of it that was recommended was this analysis on additional different types of samples. And this is where we collaborated with Karen Cush here, got samples from Stanford, and got samples from NIH, and performed the cell-free DNA analysis and compared results. And then finally, what is recommended is this third step, where to validate the analysis in an external cohort, and we've already done that as well. So just a little bit of preview. This is showing here the Stanford's work and the NIH work, and with on the inside one particular patient, and we're measuring the donor fraction, tracking this person through several months post-transplant. And you can see they're absolutely superimposable. And this is the analyzed data of the several patients that we saw and showed that the work is very comparable. And we've done that, and that there's a manuscript in press. So the first step, I would say, in the transfer of the technology and the reproducibility was done. Sequencing becomes very expensive, right? And the depth to which you sequence adds to the cost. And so we wanted to know what was the optimal sequencing depth in order to reduce, while we maintain precision and just reduce cost and time. So Sean in my lab took samples from 2 healthy people, and assigned one as recipient and one as donor, got their plasma cell-free DNA, got them in different sort of aliquots, and sequenced them to a depth of 250 million reads. And then sampled from each of these, starting with 1 million, and you can see then computing the donor fraction, and then repeated that 20 times, and then taking 5 million, 10 million, and so forth. And what we find is that the optimal sequence depth is about 10 to 15 million reads, and that actually results in quite reproducible results. And so that has resulted in considerable cost saving. The other challenge is to do this rapidly. Okay, so right now, and at the time when we did this work with Steve's, it's sort of weeks to get this kind of work back. And if we're thinking in terms of translating this to the clinic, we've got to be able to do it faster, better, and cheaper. And so what we've done is utilize various robots that take away this need for the manual preparations of the DNA libraries, automate it, and then sequencing. And it actually, this surprisingly increased the workflow tremendously, over tenfold, and reduced cost quite considerably. So that's also in place. Let's turn now to application. And I mentioned that we're very interested in the various phenotypes. I showed you the patient who came with antibody-mediated rejection and allograft vasculopathy. I've shown you the data that's to do with the poor outcomes in lung transplantation for a BOS, etc. So we wanted to see whether or not, from early readouts, there is a signal that might differentiate these phenotypes. And here it is. This is a small number of patients, but AMR is antibody-mediated rejection, ACR, and you can see the percent donor-derived DNA. And what you're seeing there is that what I interpret is that the degree of injury is significantly greater in the patients with the antibody-mediated rejection, and this is for hearts, and we've shown very similar finding for lungs. Going back to the lungs, I mentioned before, and I showed you that their outcome is extremely less, it's not as good as in heart transplant. And we are wondering whether they are being injured more so in the early phase, and thereby setting up these poor outcomes. And what you're seeing here is the donor-derived DNA over time. And what I'd like to show you is that the lung represented in blue and the hearts represented in red have a very different time decay. What you see is that the heart transplantation relatively high levels, 5%, etc., at the beginning, right there after transplant, but then rapidly decay to very low levels. On the other hand, the lung transplant have this initial decay and then a very slower course. And what we are interested to understand what is going on there, both mechanistically and perhaps even more importantly, initially, whether or not this pattern predicts these devastating outcomes in the patient. And so we've done some initial analysis, and the answer to that is probably yes. What Sean in my lab did was to take the average percent donor-derived DNA within the first few months, a month, 6 months after transplantation, and divided it up into a percentage greater than or less than 1.2. And doing so, you will see a very different outcome in the patients who have higher levels of this early donor-derived DNA compared to others. Now, these are a small number of patients and needs to be validated further to understand what's going on. But I think it's the initial observational work that will now lead us to ask other questions about this. We will externally validate this in the larger cohort of the graft samples. I showed you that is 200 patients already after a year. It will quickly increase to greater numbers, and if we can bring on more centers, we can rapidly have the answer to that work. Let's switch tacks now to talk about mitochondrial DNA and how that might be an interesting DNA for organ transplantation. So my interest in this work really came about through this paper that was done by Toby Juice and Sonia Schopfer, who were actually here in cardiovascular surgery a while ago and then went back to Hamburg. And what they showed was that these somatic nuclear transfer derived embryonic stem cells with mismatched mitochondria can actually trigger an alloimmune response. And so what they're showing here is that the mitochondria coming from the BOB-C or the BDF1 then injected into either of those animals results in rejection. And what they went on to show was that the allogeneic mitochondria even when they have identical nuclei, so now we're talking about mitochondrial DNA, nuclear DNA, despite the fact that nuclear DNA are identical, you can see these adaptive immune responses, and they are directed against the mitochondrial content. And more than that, they've shown that the mitochondrial content can trigger and bind MHC class 1 and class 2 and recognize CD8 memory cells. So I was intrigued to see this paper just as I moved to NIH, and then in the course of this, we had even de Vladimir, who is the postdoc in Steve Quabe's lab, come and help us transfer the technology of the donor-derived DNA. And while we were talking there, I said to him, Ewan, what about mitochondrial DNA? Do you think that that is something that we should be going after? And what he said was, yes, it was very important, and these are some of the reasons here. Just a little primer on mitochondrial DNA for those of you. I had to go back, by the way, and learn all this stuff again, because it's a long time since I've been following this literature. It's circular, the mitochondrial DNA. It's actually maternally inherited. I think everybody knows that. It is of bacterial origin, and this is important when we begin to think about its potential pro-inflammatory effects. has more, there are more copies per cell, of course, you know, in the nuclear, you have just 2, and it has a higher mutation rate. So it encodes just 37 genes, and they're functionally very relevant. We know all that already. So as I looked at the literature, I became more and more convinced that this might be important, and I was able to convince Ewan that this was actually also important. There is another characteristic of mitochondrial DNA, which I think you ought to know, and it's called heteroplasmy or homoplasmy. And that is this ability for homoplasmic cells to have all identical mitochondrial DNA, or on the other hand, for some of the mitochondrial DNA to have mutations and some not, and this is called heteroplasmy. And it is used by investigators to talk about the heteroplasmic frequency. So you have set up here a situation where you have more likely to have mutations, you have a pro-inflammatory situation, and I really believe that that might be enabling us to think more about it. And some of this I've already talked about. It's abundant in the plasma, a large number of variants, and the other part of it has to do with the haplotypes. And I'm sure you have seen presentations about this already, but I was really intrigued to be reminded about. Turns out there are various mitochondrial haplotypes that are predictive of ancestry. So for example, the L haplotype, L1 haplotype, very common in Africa, whereas the H haplotype, more common in Europe. And that's not by accident, because this is linked to the function of the mitochondria, whereby the need to provide heat is more important for people in the European areas than in African areas. And so it's functionally very important. But the bottom line is that there is this difference that could actually be important, and taking into consideration with the work I already shown you about
Stanford University.